right. Hello, everyone. I hope you've been doing well. Welcome to the last wet lab module. I hope you've learned a lot about wet lab, and I hope I've introduced a lot of the basic knowledge that I honestly wish I knew coming into wet lab. And I hope I've really helped you think more about how to design experiments and what parameters and features are really important to think about. You're actually running experiments at the bench, especially experiments where you're dealing with a living organism. So, and that actually leads well into the topic of our last module, which is about exploring different experimental models. So, today we'll be going over in vivo, ex vivo, in vitro. You might have heard these words, but have not, if you didn't know what they mean, then we're going to go over them. And then similarly, we can also go through, I do want to focus on animal models today because in the advanced lab, we do use animal models. We primarily use rat models, but we also work with ferrets, we work with pigs, mice. And so it's really important to understand in terms of experimental design, what kind of animals are you should use for different experiments. So let's just jump right into today's module. All right, so this is the last module and we'll be going over experimental models like I mentioned. So I guess to start, and this is something that I will collect in the Google form, but I just want you to think about what types of experimental models have you heard about? Have you heard about in vitro, in vivo, or ex vivo? What type of models did you notice in your literature reviews? Like what type of animal models, if they do have animal models, do they use cell models instead? And really, what are some disadvantages or advantages to using these models? And that's something that I hope that you can think about and you'll learn throughout this module more information about what these different models are. So first and foremost, I do want to point out, since I will be talking about a lot about animal models here, is that we do follow a strict set of animal ethics. So of course, animal models are so important to the advancement of science, but we do have to keep in mind that of course they are also animals, and we have to make sure that we're treating them with respect. And so I do want to point out that every university does have to abide by a lot of strict animal protocols. So the first and foremost, the most important thing is that animal lives are precious and we must honor their sacrifices made to advance science. A lot of really important research advancements have been made with the help of animals. So COVID vaccines, hamsters, actually a lot of hamsters being used for the COVID vaccines. And COVID, it basically stopped a worldwide epidemic, which is amazing. A lot of animals have been used and, to study genetic disorders, development, specifically flies are a really popular animal model to use in this type of research. Animals have helped us understand more about surgical procedures as well as behavioral studies. And I do want to point out as well that animals benefit from these studies. So there are a lot of vets actually that use veterinary science that actually use the results of animal studies to help them learn how to take care of animals better, actually, and how to treat different animal illnesses better. So, these sacrifices the animals have made also benefit them as well as us. So, every lab needs a strict protocol on animal care and use. Here, I've taken a screenshot of the Office of Laboratory Animal Welfare, which is based out of the National Institutes of Health. So this is a national organization that really makes sure that has a checklist for animal studies. And we actually have inspections very frequently. We actually had an inspection like 2 weeks ago from a governing body for animal care. And so, we do everything we do is we do our best to be very humane and respect the animal. So start thinking about what makes a good model organism. Uh, First off, it's important to understand that for like this example is of a rat, and I put the rat first because in the NAMS lab we primarily do use rats. We use a neonatal rat actually, so they're rats that are supposed to model neonatal babies, so within 35 days old. So you can, if we start thinking about why rats are important or are an interesting animal to use, one of the reasons why is because it's scalable. It's easy to breed rats, and can have large colonies. On top of that, many disease-linked human genes are actually expressed in the rats. Actually, I think almost all disease-linked human genes have counterparts in rats. So pinpointing these would really help researchers develop rat genetic models of human diseases. Uh, uh, Rats are also often used to study behavior psychology, so fast learners and generally do have higher level of strategy than mice. Their brains are larger than mice as well. So because of this, the animals are less timid and also more intelligent as well. And if you've ever handled mice or handled rats, you might know that rats are actually more docile than mice. They're actually more closer. I've heard them being described as closer to dogs than you might think. Actually, they're very loyal and they're very docile as well when they get to know you. So they are easier to handle than compared to mice, actually. Personally, I've dealt with both rats and mice, and I will agree with that comment. Mice actually kind of scare me because they kind of jump around. They're very aggressive in terms of like they have a lot of energy and they jump around a lot. So it kind of scares me to handle mice because I'm always nervous that they'll like jump out of their cage. But it's interesting the different behavioral aspects between a mice and a rat, even though they're both rodents. Although the rats don't think like humans, some of their brain structure actually correlates really well with more primitive elements of the human brains. And because of this, they can be used to model human behaviors. So they show more stable performance in cognitive tests. They're actually, again, like I mentioned earlier, they actually show a higher level of strategy than mice. They're actually better at maze learning than mice are, actually. And they need less training to go through performative tests like mazes than rats do. A lot of rats are actually, or sorry, the mice do. A lot of rats also are used to study addiction because they do show compulsive and addictive behaviors. So there's a lot of research that has really advanced in the addiction research field because of the use of rats. And that makes them really interesting models to work with. And again, they have similar brain structure to humans. And so it's a good model organism for the Lance lab because we work with brain. Now, I talked about rats, but actually mice are more common. I will say mice is probably the most common animal model that's used in research. A big part of this is because it's scalable. Now, I said rats are scalable, but mice are even more scalable. They have very high like breeding. It's very standardized breeding methods, and you can breed them really easily as well. So they're definitely scalable. I'll also say that due to the genome sequence being published and available in December 2002, the mouse genome has shown to be very similar to humans. They have about 75% similarity. So that's part of the reason why mice are also used a lot, just because they are scalable, they're also cheaper, and because of the genome sequence that was published, they have a lot of similar genes. And there are a lot of genetic variants that are available. A lot of researchers are interested in mice that have been engineered to be deficient in certain genes, for instance. And in that way, you can study different genetic variances and study how different genes affect the overall physiology and overall behavior. So that is one reason why a lot of mice are used. Mice are also small, they're easy to maintain. They also have a short life cycle. So because of this, they can we can monitor them over multiple generations as well, um, which is important for understanding like population-based genetics because you can have multiple generations of mice. There's, like I mentioned, mice are just a more common organism, and so because of that, there's just more published techniques on mice studies compared to any other model. So you just have a lot of information, a lot of background knowledge on mice already. Um, and many human diseases have actually have genetic similarities to mice, uh, which is interesting actually. A lot of complex diseases are that mice have, we actually share, they also share with humans, such as hypertension and atherosclerosis. Um, because they share many of these genes with us, or mice also have these diseases that we can study. So research in mice really provides us a lot of insights into genetic risk factors for studying a lot of complex diseases within humans. So they've done a lot, had a lot of contributions to our research on complex diseases. Now, this is something that you might not have actually thought about, but dogs are actually used in some research areas. So dogs are known to be man's, they're called man's best friend. And it's called that for a reason because we actually share about 84% of our DNA with dogs. So we actually share more of our DNA with dogs than we do with mice, as I showed earlier. Um, Dogs are very physiologically similar to humans, actually. So they're used in a lot of biomedical research in investigating heart and lung disease, cancer, and orthopedics, as well as aging, because their long lifespans are actually make them really great to study long-term health conditions like aging. Rats and mice don't live as long as dogs do. So dogs are really popular for aging research, actually. And on top of that, they're really cute. Of course, they're really useful for genetic studies because they suffer from similar diseases as humans. So the research that goes into, because again, we share a lot of our DNA with them. So a lot of the research that goes into studying complex diseases in dogs actually does help dogs as well. It helps. It's, again, a lot of vets have used research from dogs to really improve the way that we care for these animals. Dogs are, of course, really easy to train. Well, okay, I should say easy, but they're definitely trainable. And they're also really fast learners and they're really intelligent and smart. Dogs are also known to suffer from similar diseases to humans, such as diabetes, epilepsy, cancers, eye diseases. And the training, just going back to the training, because dogs can be trained more readily than mice or rodents or other rodents, you can train them to do a lot more behavioral studies, to a greater complexity of behavioral studies than you can for a rodent model. So yeah, they are truly a man's best friend. Now, fruit flies are, I think I mentioned this earlier, but they are an amazing model for genetics research, actually. So they're 75% of the genes that cause disease in humans are found in the fruit fly, the Drosophila, that's the scientific word for it. Drosophila are probably, out of everything that I've talked about so far, all the model organisms, Drosophila are this most cool one. You can grow Drosophila like crazy. You don't even have to try. I'm sure if you have any experience with, if you have any experience with fruit flies, you'll know that they multiply like crazy and you don't know where they come from. They just multiply. They're just good at multiplying. And you can really control their breeding rate can be controlled very easily based on just like changing the environmental temperature, for instance. So because they're scalable, they're easy to raise, they're very, very cheap. They have a short reproduction cycle. I think the reproductive cycle is like literally 8 to 14 days, so very short. you can like produce multiple generations very cheaply and very quickly. And because multiple generations can be raised, again, this makes them really great model for studying genetics at a population-wide level, where you have multiple generations of humans, and you want to understand how different genes get passed down through these generations. Fruit flies are also small. So they're about 3 millimeters long, but they're actually not so small that it can't be seen without a microscope. So you can actually still feed them even without a microscope, and that makes it easy to take care of. The fruit flies also is a simple way of creating. It's easy to generate a lot of genetic variants of fruit flies. So like I mentioned earlier, for mice, there's actually a lot of variants that are produced, like genetic engineered mice. There's a lot of strains that are already engineered, but for fruit flies, it's even easier. So you can really play around with the different genetic variants and really start to understand how different genes interact with one another, what happens when you take away a gene, what happens when you add a gene. You can play a lot with these different genetic variations with a fruit fly that you really can't do on other animal models. That's what makes them great for this type of research. And also, if you ever do take biology class in college, you'll learn about developmental biology probably, and you'll learn that fruit flies are used a lot in developmental bio research, because you can their larva is visible, and it's also a really simplistic model, so you can really see and watch the developmental stages happen. Oh yeah, anyway, fruit flies are really popular for genetics research and developmental bio research. Now, these are guinea pigs, as you can see. They're adorable. I love guinea pigs. They have very physiological similarities with humans. So actually, my very first lab experience was at an asthma lab, actually, at Oregon Health and Sciences University. And in this asthma lab, they actually studied guinea pigs are actually their model of choice, because guinea pigs have very similar respiratory features to a human. And they've actually been used as experimental animals for a really long time, believe it or not. Hence the term, if you ever heard of the term guinea pig being used in colloquial terms, that actually refers to being like an experimental subject, for instance. So if somebody says that, if your mom wants you to be a guinea pig for her new recipe, that means that she's trying to say that you will be like her little experimental test. You'll test out the recipe before she feeds it to other people. So yeah, guinea pigs have been used as experimental animals for actually a long time. And because of that, a lot of really important complex diseases have been worked on, and we've learned a lot about a lot of complex diseases with the help of these animals. One big disease that was discovered in the guinea pig is actually tuberculosis, which is a really devastating respiratory disease. And yeah, because of their similarity to humans, and because of the fact that they're also larger, it does make it easier to study more of these complex diseases and be able to gather more tissues, so we have more samples. Guinea pigs are actually also really sensitive to infections because they have very similar immune systems to humans. And so because they have a lot of similar immune system to humans, they have really similar I guess immunogenic effects and reactions to humans. So because of this, a popular research area that uses guinea pigs is allergies, like any research with allergies or respiratory diseases that are triggered by allergies. A lot of nutritional research and also hearing research fields are often used in guinea pigs. So they're actually quite more similar to a human than you would think. And importantly, their immune response is very similar to that of humans. Now, going on our list of animals, this is probably the most different one. Well, no, the Drosophila kind of was different, but this is another one uh, that isn't a mammal, and this one is a frog. So I mentioned that a lot of developmental research uses flies, but also a lot of developmental research is performed on frogs, actually, which is really interesting. Um, this is because frogs are scalable. You know, they have a bunch of eggs that you can grow and hatch, and these eggs and embryos are relatively large and robust, and they can develop in really simple salt solutions. And because they're also more like transparent, you can actually watch development, and the development can be like observed at multiple stages as well. So that's why it's really popular to study these frogs in the developmental research space. The eggs, have you ever seen frog eggs? They are opaque, but they do develop into transparent tadpoles. So it's easy to develop the embryonic development and a transformation of a tadpole to a frog. You can really watch how legs are grown, which is really cool, actually. You can really manipulate because these embryos often um, are often outside the body. You can you can really study and manipulate the development of these embryos. Um, and and these frogs actually produce eggs all year round, so they're not like you don't have to control for egg stimulation or anything. So the eggs are produced all year round. They're scalable, like I mentioned. They're also smaller in size, but not too small that you can't see them still. So it makes it easier to take care of. Um, and because they're small, they also don't take up too much space in the laboratory. And they actually also live a really long time. Frogs can live up to 30 years. So you can, again, study multiple generations. You can study aging in frogs. And all of this is really, really interesting work that has really benefited the field of aging as well as development bio. Now, this is something that you might not have seen before, but it's a really cool looking fish. It's called a zebrafish. And since the 1960s, the zebrafish has become increasingly important to scientific research. So these fish are scalable, they're really cheap to maintain. They really require minimal resources compared to the other animals that I've shown before. I'd say besides the fruit fly, all the other animals do require more resources than a zebrafish. Zebrafish are also small, so they don't take up too much space in the lab. Mating is also really easily triggered, actually. The break of daylight can actually trigger mating in zebrafish. Some other fish only lay eggs in the dark, actually. So zebrafish are actually really flexible. And because of this, it's also easier to breed them and take care of them. They grow at a really fast rate. They can develop as much as a day. They can develop as much in a day as a human embryo can develop in a month. So you can imagine that the development is really fast. Hundreds of offspring can be produced weekly. The combination of these makes them a really great model for developmental research. And I think what's also really cool is that embryos are also transparent. You can image them and actually see what's going on as they're developing, which is really cool. You can actually, researchers have used these zebrafish a lot to study the development of organs, actually, because you can see the organs develop inside the body. And what's even more cool is that every blood vessel in a living zebrafish embryo can actually be seen using just a low power microscope. So just from module 2, you'll know that there's a lot of different types of microscopy, but you could visualize blood cells in a living zebrafish with like a light microscope. So you don't need that much high tech technology to be able to image them. And I think this is the coolest thing about zebrafish, but their heart muscle actually regenerates. This is a really unique ability. So, for example, if part of their heart is removed, it could actually grow back in a matter, which is crazy. And scientists are working to find out the specific factors that are involved in this process so that we can, it can help us be able to develop ways of repairing the heart in humans who have suffered from heart failure or have suffered heart attacks, for instance. So that's a really cool aspect of zebrafish that we can really apply to human research. Okay, so second to last animal, and this is the worm. So I have to say I don't love the insects. So this and the Drosophila are my least favorite animals, but I will admit that worms, or C. elegans, as the scientific word, can be grown really cheaply, very scalable. They're so small. They can be grown in just plates, honestly, like lab culture plates. The worm is transparent, actually. They have completely transparent bodies. So you can really see, and you can image them really well, and you can also see, really follow like the behavior of individual cells through its development. So for instance, there's actually a lot of images out there that feeds, you can feed like a dye, like a fluorophore to these worms. So you can see the fluorophore travel throughout its body, which is really cool. It helps us really understand the movement and the distribution of different drugs, for instance. And actually, because of that, these C. elegans also are screened thousands of potential drugs for important diseases, actually. Just because you can treat them easily, you can really watch in real time as these drugs get distributed throughout the body. So they're really good at screening therapeutics. And overall, it's a relatively simple organism. It's not as complicated as, it's probably the least complicated of all these models that I've shown you today. And because of that, because sometimes simple is actually better, even though it's a simple organism, a lot of its molecular signals controlling its development are also found in more complex organisms like humans. So even though it's a simple organism, which in turn makes them easier to study, they also share a lot of developmental periods and developmental changes that more complex animals experience. Okay, and this is my last one. I saved this last animal because it's my favorite animal. This one is called an axolotl, if you've ever heard of them. They're actually salamanders, and they've actually been used for research for more than 200 years. I love axolotls because they're always smiley and they're really cute. But what makes these salamanders really cool is that it could actually regenerate any of its limbs, literally. Like, it can regenerate its tail, its legs, even parts of the brain and spinal cord, which is crazy. It can regenerate damaged parts of its heart and its lungs. Pretty much almost any part of its body, it can regenerate. So you can imagine that these creatures are really, really important for our understanding of regenerative medicine. Understanding the cellular and genetic mechanisms by which axolotls regenerate tissues, you can imagine, can have really strong clinical significance for treating human trauma, disease, and aging. So, and axolotls are also really unique. Okay, the regenerative capability already makes it unique already, but what's even cooler is that they actually maintain juvenile external characteristics all throughout life, which makes them an even better candidate for aging and developmental research. So, just imagine maintaining the appearance of a teenager when you're like 80, right? So, which is crazy. So unlike many amphibians, axolotls maintain their juvenile external characteristics because they actually never undergo metamorphosis and transition to land. So they stay in the water. So unlike frogs, they don't go through this transformation. Because of that, they remain in this juvenile state for their entire life, which is crazy. And they actually don't get cancer either. So that's a whole other research area that makes them really great candidates for using as animal models. And because they live in the water and they haven't gone through the transformations of other amphibians, having an aquatic environment makes them a really good research model for toxins in various bodies of water or for studying environmental impact. So I've been talking a lot about biomedical research, but axolotls are actually used outside of biomedical research and like waste, water quality research, water control, which is really cool. Okay, now because that's all the animal models I will talk to you about today. Again, if you have any questions about any of these animal models, please feel free to Slack me. I've worked with my fair share of animal models, but obviously I haven't worked with all of them. But I do think it's really important to learn about the contributions that each animal plays in the history of human medicine and human advancement of technology. So yeah, do your own research and really think about what types of animals, whenever you're reading a paper, understand about like why the researchers chose the animals that they did and what types of animals have really advanced specific fields throughout the years. Oh, right. Now, holding off to talk about animals, we're going to talk about Latin words. So there's these words that you might have heard about, but you might not know what they mean. The first is in vitro. So in vitro is Latin for in the lab. So this means that in vitro work takes place or is performed in a test tube, a culture dish, basically anywhere like outside of a living organism. So you're growing, like for instance, growing cells on a culture dish. So that's in vitro work. And usually when you're growing cells in vitro, you're usually growing just a couple of layers, usually in monoculture. And just keep in mind that it's limited in scope because the cells grown on a dish, like on glass, is not very physiologically similar to that you would find in the body. Now, you might have also heard of the term in vivo, which is Latin for in the living. So this is any tests, experiments, or procedures that researchers perform in or on a whole living organism, such as a person, an animal, or even a plant. So all of any research that's done on a living animal, for example, the animals that I just showed you about, you would call that research in vivo research. So like usually this would involve some sort of surgical procedure when working with these animals. And lastly, you have ex vivo, which is Latin for outside of the body. And this is a procedure in which an organ, cells, or tissue are taken outside of the body and then returned, like to outside the body for treatment and then returned to the living body. So you're like taking some tissue and taking it outside the body. And we do ex vivo work in the NANS lab. We also do in vitro as well. We do grow cells in culture dishes, but we do a lot of our work in ex vivo work. So we're taking a brain, we're extracting the brain, and we're culturing the brain outside of the body. And you'll actually see a video that's actually in combination with this module to see what that process of brain extraction, brain slicing, and preparing these ex vivo models is like. So I encourage you to watch that. It's really cool experimental work. So as I mentioned, my goal is to really have you think about why, like how certain models are interesting and what types of models should you use when designing your own experiment. As you can imagine, there's advantages and disadvantages to a lot of different models. So for in vitro, it's less complex. You're literally growing something on a plate. So it's less complex, less expensive. It's more flexible and scalable, and there's just less regulation because you're generally just growing cells on a plate. Disadvantages is that what I mentioned before, because you're growing it on a plate, it's not very biologically relevant. Like it doesn't really match environmentally. It doesn't, the cells grown on a plate is very different from the cells actually living and growing on inside of your body. So it doesn't really capture as well the multiple cell type communication. As I mentioned in the second module, tissues are made out of layers of cells, right? And when you're growing cells on a plate, it's usually just a monoculture, so it's one layer. And you don't really capture that complex layering of cells that you would in like tissue. In vivo work, the advantages are that this is the most like the closest you can get to biological conditions because you're actually performing them within a biological organism. It's also more clinically relevant and translatable, and you're able to study effects of drugs on the body as well. Now, in vivo work, even though it's the closest you can get to matching what happens in the body, it's also expensive because you're actually using you're using animals. It's because it's expensive, it's less scalable, there's stricter regulations. Experiments are also longer because cells proliferate really fast, but animals take a while to grow and breed. So they're also slower. Lastly, ex vivo. Ex vivo is really a balance. It's kind of in between in vitro and in vivo. So it's less expensive than in vivo, but it's also more clinically relevant than in vitro. And it's also scalable because, for example, for the Wetlab, when we take brains and we slice them up, we can actually, one brain actually provides us a lot of different slices. And that's the benefit of using this type of work. So you can just, from one brain, we can get a lot of slices and we can reduce the number of animals that are actually necessary to perform our experiments. Disadvantages is that it's less clinically relevant, again, because it's still outside the body, so it doesn't completely replicate what goes on inside your body. And the cultures can't be maintained for long term because you're literally taking a tissue out and growing it outside of the body. And tissues don't grow really well outside the body. We haven't really figured out yet how to grow tissues outside of the body because tissues have multiple layers. And each layer needs nutrients, as you can imagine. And it's really hard to feed layers of cells all the nutrients they need. Oftentimes, the center of the tissue starts dying because they're not getting the nutrients that they need to survive. So cultures, these ex vivo cultures really can't be maintained long term, at least yet. So again, like I mentioned, I do have a video where I show you how we prepare our ex vivo brain slicing in the lab. So you can watch me in action, slicing brains, and you can really see how these ex vivo models are prepared. Okay, and you've reached the end of the third module. Now you've known probably way more about these animals than you did before. And I hope you are able to take away from this really just understanding how different animals have really benefited research and how different models provide different advantages and disadvantages to research. Now, when you go out into the world and develop your own experiments, you're going to have to face one of the very first questions is probably what type of model you're going to use for your research, for your applications. And it really just depends on what you want to do. So these are just things that you'll think about that I will also add in the quiz or not the quiz, but like the Google form post module activity. But think about what type of model would you use for testing out medications? What type of models would you use when you're trying to figure out how different medications move throughout the body? And what about figuring out if nanoparticles are toxic to cells? What type of model would you use then? Or how about thinking about running a clinical trial on a new vaccine? What type of animal models would you use to study this? So these are questions that researchers have in the lab every day. And every time you create a new experiment, you have to ask yourself what type of models you need. Like, do you really need to use a guinea pig for what you can do with a fruit fly, right? So these types of questions are really, and considerations are important when you design your own experiment. Yeah, and with that, I hope you learned something about different experimental models in biology, different animals, why we use the animals that we use. So you can go ahead and be more critical readers and thinkers when you're reading these different research from the journal articles that we will be sharing throughout this module and throughout this program for you. And with that, please feel free to ask me any questions from Slack. I'm happy to help, and I will see you later. I hope you all have a good rest of your day, and please fill out the post-module activities. Bye.